make sure that we're not rushing through stuff today, and then we also have some time to do some review questions. So I plan that Monday, that's what we'll spend most of the time doing. And then Wednesday we'll start Ebola. But again, that's not on the next exam. So the cutoff is whatever we finish up on Monday. In-class portion, April the 6th, which is that Friday after Easter. So it's 2 weeks from today. The take-home, my goal is to get that to you that Wednesday after Easter and give you a full week and a half to work on it. Not that I'm imagining it to be any harder, but just I want to make sure that I'm respectful of your guys' time and give you adequate time to work on it. What's that?
Can I assign a notebook?
Oh, crud.
I don't know.
Oh my gosh, Grace. Just falling apart.
Looks like my life.
Yeah.
I brought him up today.
Welcome to the club. Always looking at the positive. So that's kind of a heads up. I'll admit that I don't have all the Ebola slides done yet, and at the latest I'll get stuff posted on Monday. I'll let you know if there's going to be updates or not. But actually on Wednesday, what we're going to cover is we're actually going to cover epidemiology. We're going to cover some of the outbreaks that have occurred, and we're going to talk about using Ebola as a focus, talk about what's the difference between a pandemic and an epidemic. And we'll talk about a couple of just features of epidemiology because they relate really well to Ebola. So that's kind of a primer for what's coming up. I still haven't gotten sequences back. I'm going to email them today to find out what's going, how everything's going. But I think we have enough money banked that we can actually get images of every single one of the phages that we've sequenced as well. So those are heading off on Monday. And once I get the pictures, you can use them for the posters or whatever else you want to. All right. Did anybody? I know one group got an acceptance letter for this. Did anybody else yet? Oh, looks like Grace and Tori are the only ones presenting so far at Edgewood Engage. So, wouldn't that be sad if you guys were the only ones?
We could all come together and do it.
We could. All right. Last thing. I know some of you are aware of this, but on Tuesday night, 6 o'clock, I am doing a 2-hour talk on my family. So, if any of you want to come, it's in Washburn. It's free.
What is that?
Tuesday at 6.
I have my night class, but...
Skip your night class. I give you the full ability to do so. So, feel free to come. What's that, Sabrina?
Could it be extra credit?
No. Well, of course it is.
No, we're going to charge $5 at the door.
Maybe I will. Maybe I will. I'll tell you what, that price of that hormone therapy is not cheap, so, alright. No extra credit on this one.
I was just kidding.
Okay, well that's okay. I don't want to exploit my son for extra credit, that's all. I'll exploit him for other things, but, alright. So let's start off where we finished up on Wednesday. So we talked about what we had finished up with on Wednesdays. We talked about E6 and E7, their roles in regulating for E6, P53, for E7, binding to E2F and inhibiting the binding of RB to E2F. And we talked about, hey, what would a Western blot look like with each of these four different, or in four different conditions. One with just an empty plasmid transfected into cells, one with E6 transfected, one with E7, or one with both of them at the same time. Now we took a look at what would hypothetical results look like for that Western blot. Let's talk about reporter assays real quick though. So let's go back to this idea of a reporter assay. And I'll kind of create the setup for it. This one says you're going to use GFP as an indicator of transcription. Well, what's really nice about that is you can actually use fluorescent spectrophotometers to measure how much GFP is being given off. So you can make this a quantitative experiment. And essentially, if you were to, what we would want to do is transfect into those cells an additional plasmid that contains a promoter that can be bound by E2F and the gene being regulated, instead of being one of the typical E2F genes, in this case would be green fluorescent protein. Okay. So what I want you to do is take, let's start with about 3-4 minutes of, again, each of those 4 conditions, without E6 and E7, with one or the other, or with both. What would you expect the expression of GFP to be from these promoters? And let's just make this very simple. Don't worry about shades of gray. Just, is it on, or is GFP being expressed, or is it not being expressed in each of those conditions? All right. Go ahead and talk it out with somebody else. All right, let's talk this one out. So, erase some of this. All right, so here's our promoter. Again, 4 conditions. Nothing, just E6, just E7, or both. Oops. Without either of them, what do we expect to see expression from this promoter? Maybe a little. I mean, if we're talking shades of gray, yeah, maybe some. Just naturally, some cells are going to be undergoing cell division, right? If we were talking without shades of gray, I'd say no, there's not going to be any, right? What about with E6? Why? What is the function of E6 again? Or what's the mechanism, I guess, that would lead to expression of GFP here? So walk me through the steps. Good. So then the cyclin and cyclin-dependent kinase is active, and what do they do, Shania? Good. Good. Perfect. Exactly. Great. So yeah, we'd expect to see some there. What about with E7 only? Probably. And Sabrina and I were talking about, well, would it be as much as E6? And you could make an argument here that maybe not, because one of the things that we said is E7 alone isn't sufficient to cause cancer or to transform cells. One of the downsides is P53 is still active. Okay? So, but remember here, the mechanism with E7 is what? How does E7 function? Binds to E2F. So it's independent of the cyclins and cyclin-dependent kinases. Okay? And with both of them, this is probably where we'd see, if we were looking at shades of gray, this is probably where we'd see the highest expression, because now you're inhibiting 2 inhibitory pathways. Okay? Any questions on this kind of experiment? Okay. Folks feeling comfortable with this? Starting to click a little bit? I have to say, Brenda commented after class that she was really impressed with what you did. So, I'm proud of yourselves. All right, let's move ahead. So, what I want to finish up covering today are a couple of things. One, the replication process, and then let's try to get ourselves talking a little bit about HPV-caused cancers. So obviously, again, if we take a step back and look at this from a holistic view, E6 and E7, their role is to promote S phase. That means that DNA polymerase is going to be transcribed and translated. That doesn't tell us, though, how does the virus actually recruit DNA polymerase to the HPV genome to support replication. And this is where E2 comes back into play, and also another one of the viral proteins known as E1. So one of the things you may remember, E2 is a transcription factor, it controls splicing, it also binds to regions within the URR. Okay? And that again is that promoter region. It also contains the origin of replication. So here is represented 3 binding sites for E2 within that URR or that LCR, the long control region. What E2 does is it recruits E1 So it will bring E1 to the origin, and E1 is a helicase. And like all helicases, its role is to begin separating the hydrogen bonds that hold base pairs together. E1 will kind of oligomerize, meaning that it will bind to itself. And at this point, E1 has docking sites for DNA polymerase. And so this is the actual recruitment step. So once E1 has been bound to the origin of replication, and now the host DNA polymerase can come to that origin of replication and begin replication in both directions. And at that point, it's just like regular eukaryotic replication or even prokaryotic replication in the ways that you've talked about in other classes. Okay? So that's about the depth that we're going to cover that process. Shania.
What was the purpose of it binding to itself or oligomerize?
I think that, I don't know the exact purpose of it. I think that it's required for it to have its helicase function, but also it's required for it to bind to the DNA polymerase as well. Oh, go ahead, Grace.
So E2 brings in E1.
Yeah.
So does E2 have to bind to all three of those sites, or?
That's a good question. I don't know the answer to that. But that would be, how would you test that? So how would you test that? If you wanted to know, are all three of these binding sites required for E2 to recruit E1, what would you do? Mutate one or more of them? So maybe start with a plasmid that is not HPV at all, right? It just contains this or this URR. Mutate one of these sites so that E2 can't bind to it and see if E1 is still recruited. Or mutate two of the sites and see if E1 is still recruited. I think that would probably be the best way to determine it. Are all three required? I have no clue. I don't know. I've never come across that in the literature.
And that's a URR?
Yeah, that's the URR.
Okay.
That's a really good question. Okay. One of the things that we mentioned is in the context of the viral life cycle, when cells undergo division with HPV ensures that its genomes will be equally partitioned or equally divided to each of the daughter cells. And so there has to be a mechanism that ensures for this to take place. The mechanism involves a human protein known as BRD4, and E2 will bind to BRD4. And we'll talk about the molecular mechanism behind this in a minute. But let me show you the importance of this. So this is an image from a paper in 2004. What they are looking at are what's known as C127 cells. And instead of using human papillomavirus, they're using bovine papillomavirus, so closely related member. And they are taking these cells and so these would be normal. These would be one that have also been transfected with an empty vector. And this last one over here are ones that have been transfected with a plasmid expressing just the carboxy terminal domain of BRD4. BRD4 is a protein that has two domains, and I'll draw it out. One of those domains of BRD4 helps it to bind to chromosomes, human chromosomes. And that's what we would call the N-terminal domain, or amino-terminal domain. There's a linker, and then there's this carboxy terminal domain. That carboxy terminal domain is going to bind to E2. And where does E2 bind? URRs. So E2 will associate with the HPV genome, allowing for the HPV genome to basically piggyback on host chromosomes as they're being pulled to opposite ends of the cell during mitosis.
Can you repeat that?
Yeah. So the BRD4 protein again will bind to host chromosomes, and that's what this teal line represents here. And as this is being pulled to one of the ends of the cell during mitosis, BRD4 will be bound to E2. E2 will also be bound to the HPV genome at the same time. So basically, the genome is being pulled along with the chromosomes to daughter cells. Now, the importance of this is that this helps to maintain a pool of cells that have HPV in it. And the lack of interaction between BRD4 and E2 will lead to a loss in, I guess you could say, infection efficiency. Doesn't allow for the infection to spread as well. And it may be a little bit hard to tell in this image, but this is what we call a focus assay. So remember plaque assays that we talked about a while back? What is a plaque?
Lice bacteria.
Yeah, lice. If we're talking about bacteriophage, it's an area of lice bacteria. We also saw other examples like norovirus, where they'll measure plaques or areas where the virus has killed the host cells. Foci, which is the plural of focus, is a little bit different. These are areas of growth. And this fits really well with what we know about HPV. What it does is it promotes the growth of cells. So in this case, you're doing a very similar type of assay as you would do with plaque assays. You're staining all of your cells with some sort of general stain, and instead of looking for areas where there's no growth, you're looking for areas where there is growth, or an increase in growth. So, in other words, you're counting all these blue dots here to indicate focus, or foci. And so that's what they measure here. So, number of foci per 1 microgram of bovine papilloma virus that's being added. In C127 cells, here are 3 experiments. Here's the average. Here's the standard deviation. So they get about 150 foci. Put in just the empty vector, they still get about 136. Now put in this version of BRD4 that just contains the carboxy terminal domain. Goes down to 10, 9. Why? So what's the mechanism for this? Go ahead, Grace.
Well, it doesn't bind to the HPV genome then, right?
Well, it's got the carboxy terminal domain, so it's still binding to the HPV genome. So you would have, in this last condition, you've got the following. Chromosome. You've also got, from the cell, full-length BRD4 as well. So what's happening?
It doesn't bind to the chromosomes then.
Yeah. In other words, what we've done is there's a competition between E2 binding to this just carboxy terminal domain of BRD4 or binding to the full-length BRD4. And what we're saying is that this greatly reduces the efficiency with which those genomes are segregated to daughter cells. Okay. So it's basically, in some ways, it's like neutralizing the infection. Go ahead, Shania.
So does E2 have a preference for just binding like the carboxy one, or is it just that they're both equally likely to bind? It's just...
I think it's the latter, that they're both equally likely to bind. And I would bet that as you increase the amount of plasmid that you're transfecting into these cells, that's going to decrease the amount of foci that form. I think that's probably what's happening here. That's a good question. I'm going to skip the next slide. It's some immunofluorescence, but I just don't want to... I don't think that it's that important for us to cover. But here again is that model. Sabrina.
So, when it connects to the chromosome and that's happening during the differentiation of like.
It would happen during the differentiation, but remember one of the things, I'm going to go back a few slides real quick. One of the things that we said is during the course of an infection, HPV maintains itself actually in these basal cells as well. So as these divide, we think that some of these basal epithelial cells are actually what we would call stem cells. And stem cells have the properties of they are self-proliferating, they are self-renewing. They will produce two daughter cells, but one of those daughter cells begins differentiation, whereas the other one remains a stem cell. And so what this does is during this division process, like they're showing here, as this one cell becomes two, you would get HPV genomes to be replicated. One would stay, or one set of them, I guess, would stay in the basal membrane, and the others would begin through the process of differentiation. So really where we see this BRD4 interaction being important is early on and up through here. At the point where we start to get late gene expression, we actually get a lot of replication of genomes at this point. So there's different mechanisms that are controlling this proliferation of HPV genome replication at these later points. Does that help?
Can I ask one
Yeah, go ahead, Grace.
You said this previously that it doesn't like pass the basal membrane. Why is that?
So, like, why doesn't it go through this basal membrane
Yeah.
here? I think that I suppose the basic reason would be, well, what prevents a virus from being able to infect certain cell types?
The receptor.
That would be my argument.
Okay.
The main receptor is within this basal membrane right here. It's heparin sulfate. So that would help it to bind to that basement membrane, and that's why we think there's got to be a secondary receptor that helps it to bind to these epithelial cells as well. And if it gets past this, now you're into the dermis and eventually also into basically blood vessels as well. And those probably don't support replication.
Okay.
Yeah, it's a good question. Alright, so here is that model then. This is just showing human chromosomes wrapped around histones, BRD4 bound to those histones actually, and then we've got E2 being bound to BRD4 and carrying along with it then the HPV genome. Okay, so that's the basic mechanism by which we think that this occurs. There's been a lot of argument over this because most of the work for this type on BRD4 has actually been done with bovine papillomavirus, not human papillomavirus. So there's still some controversy whether this mechanism holds in HPV as well. Shania.
So I don't know if this question makes sense, but like chromosomes can have like euchromatin or heterochromatin, like
Yeah.
how tightly it's bound. Does that affect how well BRD4 can connect to the histones?
That's a really good question. Well, it has to be able to bind to the euchromatin because that's the condensed form that we would find during cell division, right? But whether it can also bind a heterochromatin, I don't know. That's a really good question. I don't know if that has an effect on it or not. Yeah, great question. You should investigate that. Sabrina.
So is this happening like all over the chromosome?
Yeah, we could argue that it might be happening all over the chromosome. Now, it's not, and the one thing that I caution with here is I'm not exactly sure how many copies of the genome exist in these cells during the replication process. I don't know if those studies have been done. I only can take from what I know about EBV, and in EBV cell lines, you can have 20 to 30 copies of the genome in a host cell at any given time. That EBV also has a mechanism to ensure that daughter cells are that daughter cells get a roughly equal number of chromosomes as they divide. KSHV, on the other hand, completely random. You can have 30 genomes, and one cell might get 2, the other might get 28. It's really weird.
So like if you have 20 HPV genomes, it's going to be like 10 and 10.
Yes. Yes. That is the design. Is it perfect? Probably not. I'm going to bet that on average that's what happens, but you might get times where it's like 11 and 9 or something like that.
And then the genomes that stay in like the stem cell layer, they're like just maintained.
Yeah.
They do the same thing.
Yeah. So that's why for like for a wart or something, what's the treatment for a wart? Freeze them,
I don't know.
cut them out, and cauterize it. I mean, like, you have to kill those stem cells to ensure that it doesn't come back, basically. That's the argument, at least. Annie?
What experiment do you do to determine how many copies of a number are in the cell?
What would you use to tag DNA? We've actually talked about an experiment. Fish would probably be the best way. But if you wanted to track them in live time, fish doesn't work. You have to use some sort of modified version of the genome, and I'll speak from experience on this one. I didn't do this, but what some of my friends in graduate school would do is we had this plasmid that was part of the EBV genome, and it was engineered to contain copies of the lac operator. And so it had like 5 or 6 or more copies of that. And then these cells were also had a modified version of LAC-I that had RFP on it. And so if you have good enough resolution, when LAC-I is bound to the chromosome, you can see the RFP. And as the RFP, or as these genomes are separating, you can actually watch the movement of RFP to those daughter cells.
Okay. And then you just quantify it.
Then you just quantify it, yeah. And to quantify it, actually, what they ended up doing instead of like counting by hand, so my grad school advisor's son he majored in astrophysics, I think, as an undergraduate. He basically wrote a computer program that they use for like identifying stars, to count how many copies of the genome there were and like the intensity of it.
So for fish, can you just count as one dot?
Yes, you can. Yes, so it does tell you whether it's there or not, and it wouldn't allow you to necessarily differentiate two that are co-localized. But if they are, for example, if you have a cell that's not undergoing division, so one where you've got a diffuse nuclear staining in the nucleus, you'd be able to see those individual dots. Yeah, that's a good question. All right, cancer. Here are all the HPV-caused cancers, or at least the most common ones. Obviously, everybody knows cervical cancer is caused by HPV. There are others as well. So vaginal cancer, vulva cancer, anal cancer in both males and females, rectum cancer in both males and females, and then oropharynx, which basically is sometimes referred to as head and neck cancers. I was looking at this yesterday and I was doing some quick statistics on it. This is the most recent average number of cases. They've broken this down by those that are caused by HPV 16 and 18, and as you can see, that causes the vast majority of the cases in each of these. And then there are other what we call high-risk ones that are 31, 33, 45, 52, 58. Those cause some additional cases of these cancers. If I did my numbers correctly, and I forgot my sheet for this, roughly 31,500 cases per year are caused by HPV. So that would include even this light blue bar here. Okay. Of those, 92% of them are preventable with the most recent version of HPV vaccines. And the most recent versions that are out now contain 16, 18, these other 5 and then two other common ones that cause the majority of warts that do not lead to cancer. Alright. So that's just kind of some basic statistics here. I mean, obviously, cervical cancer has been the big push, but given the number of cases in males as well, this is what's kind of led to this push of males should be protected as well, but also from the viewpoint of male can infect female and vice versa, so we want to prevent passage of HPV between individuals as well. I just put this list up here. These are some common characteristics of what we call transformed cells. Now, a transformed cell is not necessarily a cancer cell. Transformed in the bacterial world means you're putting DNA into the cell. In eukaryotic world, in human cells, when we say transformed, what we mean is that the cell has gone from a point where it can only undergo division so many times to now where it can oftentimes undergo division indefinitely in cell culture. And transformed usually, actually always, is referring to the ability of cells to grow uncontrollably, basically, in cell culture. I put this up in part because many of these characteristics of transformed cells are characteristics that we find in cancer cells as well. So, for example, formation of foci, which we just talked about, HPV will induce. Loss of contact inhibition, meaning that the cells begin to grow on top of each other. That is a characteristic of foci formation. The ability to grow when suspended in auger. Most often, human cells need to be flat on some sort of surface. They need to have contact. Imagine them as Linus with his security blanket. Okay? Without that, they don't grow. They're immortalized. They divide indefinitely. Divide and grow more rapidly than untransformed cells. For example, HeLa cells, which are the most abundant of all transformed cells that we know, replicate faster than once every 24 hours, which is super fast for human cells. Loss of cell cycle control. In particular, oftentimes we find no G0, but cells going straight from G1 into S. And then some of these transformed cells may form tumors when injected into susceptible hosts. And oftentimes the susceptible hosts are immune-compromised mice. That's the most common model. Sabrina.
So is it's so fast replicating because HPV is pushing and it keeps pushing into that space?
That's, in the case of HeLa cells, yes, that's exactly what's happening. There are other examples as well. So there's a cell line known as 293T, and they are pushed by a virus known as simian virus 40, and it encodes what's known as a large T antigen, which is where the T and 293T come from. They grow very well as well, but they are being promoted through G1S checkpoint. And oftentimes, because of this lack of cell cycle control, what you find is these transformed cells develop a number of different mutations. So HeLa cells, for example, one of the phenotypes of them is that they're polyploidy. They contain more than 2 copies of most of their genomes. Did you have a question, Michaela? Or Annie? Oh, Shania.
So what you said, like, they're called just transformed cells, aren't cancer cells. So what's like the defining characteristic once they're called like a cancer cell? Then?
Typically, we think of a cancer cell as one that causes a disease in a human. The transformed oftentimes will refer to, like, if we have a cell line and we want to allow for it to grow for the rest of its life, a transformed cell would be one where we put in like a virus or something to ensure that it continues to grow past its limit. And what I mean by limit is most cells in cell culture will only go through 50 generations. So if you were to take human foreskin fibroblasts, which is a common cell line, or common cells that are used, they'll go through 50 generations and then they stop. They're still living, but they go into what we call a quiescent state. What?
That was in like the reference paper
Okay.
for our critical thinking, that word.
Yeah. So they go into a quiescent state, or apoptosis. Or sometimes the term that's used is anoikis, but they'll go into this state where they're still living, but they're no longer dividing. That's because what happens typically in a cell, as it's undergoing division, let's imagine this is a chromosome real quick. What's on the end of human chromosomes? Telomeres. What happens to those as a cell undergoes division? They shorten, and at some point they become so short that any further any more replication would lead to the loss of genetic information. The process in human foreskin fibroblasts, that number of times that it can replicate is about 50 times. Beyond that, these become too short, and the cell, instead of continuing to replicate, which would lead potentially to loss of genomic information and transformation, it goes into this quiescent state where it lives out its life but no longer replicates.
How do you spell quiet?
I can't remember if that C is there or not, but I think that's how you spell it.
So how does HPV like go about like manipulating?
Damn, that's a good question. That's a great question. There's a couple of things that it does.
How does it manipulate the telomeres to like make them that short? Because then you could live forever, right?
Yeah.
And you could do this for the whole body.
So in stem cells always have telomerase active, right? And that's the enzyme that continues to increase these. I'm writing down one more term real quick, Hayflick limit. This is the point at which the telomeres become so short that the cell stops replicating.
That's amazing.
Yeah, named after an individual who identified it, I think. So, Shania, how would it do it? What could be a mechanism?
You'd have to have telomerase like all the time.
Yeah, I'm just going to skip ahead real quick. E6 and E7 do that.
Oh.
So E6 and E7 will ensure that that enzyme is expressed at all times, and this is in part the reason why then HPV positive cells, at least those that we can grow in tissue culture, can continue to grow. Okay, so they can maintain the length of those telomeres. So E6, and remember its binding partner, that ubiquitin ligase that helps to degrade P53, well, they have another target. It's known as NFX1. It degrades it. NFX1's normal function is to repress the transcription of telomerase. So by blocking that repression, you get expression of telomerase from E6.
What does NFX1 do?
It is a transcriptional repressor, so it's a transcription factor, and it blocks the transcription of telomerase. E6 also will bind to telomerase and enhance its activity, so that's the second mechanism. And then the third involves E7, where E7's ability to activate E2F drives telomerase expression as well. So you have blocking a negative effect, and then two positive effects on that enzyme. Freaking sweet, right? I'll just pause and make sure everybody's got what they need from this before I move ahead.
Can you go over what the Hayflick limit is again?
Yeah, the Hayflick limit is just this idea, again, that you have a number of replications that a cell can undergo before those telomeres become too short. and at that point at which they become too short, or that those number of cycles, Grace, is known as that Hayflick limit. Sabrina.
So once they reach that limit, would you say like that's when they're going to G0?
Yeah, or like permanently into G0 at that point, exactly. Unless there's some sort of transformative effect, like activation of telomerase.
So like if people have like nerve damage, could you somehow like put E6 and E7 into the, somehow like insert it into like the neuron
That's an interesting question.
to make like the nerves go again?
That's a really interesting question. I don't know.
Okay.
You've got 2 projects to do now. I'd be interested to see if that would work, or if anybody has proposed something like that to restore replication of neurons. It's a really interesting idea. I just wanted to show this real quick to give you guys a couple of ideas of what these foci look like and what do we mean by like loss of contact inhibition or changing of these cells. So here's an experiment. The first virus ever found to do this, to induce the formation of tumors, was known as Rous sarcoma virus. It actually is the Rous sarcoma is a tumor in chickens. Rous sarcoma virus is a retrovirus. And so this is just showing the formation of a foci on the left over, or right over here then. What we're looking at, normal cells, and then cells that have formed foci. And down here, kind of a zoomed in version, and you can look at the left and right images on the bottom there. The left is what we would typically see for most cells, that they have very tight junctions between them, that they form a really nice thin monolayer, and they're flat along the surface. Over here, they've lost that morphology. They have more space between them. Some are growing on top of others. Those are transformed cells. Okay. And we can even sometimes, this would be a cytopathic effect. This is actually a cytopathic effect that we would see here. So to link this then to cancer real quick, we went over some of those characteristics of transformed cells. Here are some of the characteristics of cancer cells. This is not a complete list, but we see increase in expression of what we call oncogenes. These are genes that promote cell cycling. They, where we said tumor suppressors are involved in blocking cell cycling, oncogenes have the exact opposite effect. They activate cell cycling. So E6 and E7, viral oncogenes. We might find suppressed or repressed tumor suppressor function as well. Like transformed cells, these cells can grow uncontrollably. They now can grow independently of growth factors. And we see this again with E6 and E7. They can overcome the activation of cyclins that's required from, for example, we talked about on Wednesday, epidermal growth factor, binding to the epidermal growth factor receptor to activate cyclins. E6 and E7 can overcome that. Bypass this term, I won't say it, but repression of P53 helps to do that. Angiogenesis is the ability to induce the formation of blood vessels. Once a tumor becomes larger than 1 millimeter in diameter, it needs to induce the formation of blood vessels to support those cells in the middle. Otherwise, they undergo necrosis and die. And then oftentimes, they're avoiding immune surveillance, avoiding detection by T cells, or B cells, or NK cells, etc. And Jonah asked the question on how do these overcome NK cells. I think I figured out the answer to it, but I'm going to wait until he gets here whenever. He's probably prepping for this party of his. We'll talk about it on Monday. All right, I'll leave with this and we'll finish up our conversation. I'm going to just show the next couple of slides real quick. In HPV-caused cancers, this is a dead end for the virus. somehow the virus has actually integrated its genome into the host genome. This should not happen with HPV. It is not a part of its normal life cycle. And when it happens, there's no way to get that genome out. So some sort of recombination event has occurred which allows for the genome to be integrated into the host genome. And typically what we find are the only two genes that are being expressed in these cancers are E6 and E7. Or at least those are the predominant ones. And so at this point, somebody who has, for example, cervical cancer, they wouldn't be producing more viral particles. At that point, it's just E6 and E7 driving proliferation, in addition to other mutations from the host cell that are contributing to the cervical cancer. I'd been thinking about how to incorporate this image real quick, but this is looking at epithelial cells. The dotted line represents the basal membrane, and this is looking at a high-risk HPV infection, HPV 16, versus one that's known to cause the majority of warts, which is HPV 11. This is what we consider a low risk. What you're looking at here is immunofluorescence of this tissue with three different things being stained. Green represents the E4 protein, and this is one that's going to be expressed in those upper keratinocytes. Again, E4's role is to disrupt intermediate filaments to allow for the virus to escape, so that fits with what we know about it. The blue represents DNA, and then this red color that we see is MCM2. And MCM2, just for the purposes of this conversation, it's involved in cellular replication. It's a sign that these cells are in S phase. Now, if you were to look at this, the biggest point to make is that the amount of staining that we observe with high-risk strains is much more prominent than what we find with those low-risk strains. So what that says is these high-risk strains have the ability, and the abnormal ability, to promote cell cycling, even within those most basal of epithelial cells compared to the low risk. The takeaway point, high risk have a greater ability to promote cellular growth. And a lot of investigation is going into, well, why is that? What are the genetic differences of the virus that allow for that to happen?